Joe. And I'd further like to thank Joe and Susan, who I don't see, but they kind of got my start in primatology back in 2003 when I was a field assistant at their site in Lomas Barbadal with the white-faced capuchins. So thank you guys, and thank you again for inviting me to speak today. I'd just like to start out saying that I'm trained in anthropology, and so this kind of stretches anthropology as a discipline and goes into a few others, and I'm not sure what all of our disciplines are, but this is kind of one of the first times I'm presenting this work, so if you have any questions or comments as I go along, please feel free to interrupt. Think of this more as a seminar rather than me just talking at you, because I'm going to be presenting this next week at the AAPA. It's in abbreviated form, so any questions or concerns would be great. And so, yes. So today I'll be talking about how, with colleagues, including Lynn, who just came in, we're trying to develop a model for the genomics of spontaneous obesogenic growth in a non-human primate model, one that we can possibly extend to the wild, which I'll get to towards the end of the talk. And what I mean by spontaneous obesogenic growth, I mean that this is obesogenic growth, or becoming obese, that occurs without dietary modification. Because there are a lot of dietary modification studies, and in this case we're just looking at natural variation within these primates. So as I'm sure we're all aware from the news and various other sources, obesity is an increasing problem, and I just realized that this is on mute, and no, it's not, okay. Obesity is an epidemic of worldwide proportions, okay? In the United States alone, about 30% of the adult population is obese as of 2 years ago, according to the CDC. And more than that, about 17% of US children are obese. This has been increasing over the years, although there was a slight decline last year, but there's no reason to think that that decline is going to be persistent. And this obesogenic growth is actually common not only to humans, but also to most anthropogenically impacted animals, including lab animals, farm animals, etc. Across all of these domains, obesity is increasing with the years, and we don't really have a good idea about why this might be occurring. A lot of the studies that we've looked at have looked at adult obesity and trying to figure out what the ontogeny is of that, but very few studies have actually looked developmentally at what's happening in humans and in other species. Obviously in humans, looking at the development of obesity is complicated by participation, by consent, by people reporting diets accurately, by people sticking to diets, etc. But from the few studies that we do have, we do know that maternal obesity has a definite impact. So if a mother is obese when that child is gestating, they're more likely to be obese during development. We also know that there appear to be critical periods during ontogeny, during which time if a child is obese, then they're more likely to become increasingly obese as they age. And we know that the ontogeny of obesity appears to be pleiotropic and genetically controlled. There's a large genetic signal in these small-scale studies on humans, but we still don't have a good idea of what's going on systemically throughout development and how this results in an adult obesity phenotype. So in looking for these genetic factors that are controlling for obesity, we've attempted a number of things. Genome-wide association studies is probably what most of us are familiar with nowadays. They've been really successful in mapping common variants for obesity. These usually only explain a small proportion of phenotypic variation, however, and I'll go over that in just a moment. And we often need extremely large sample sizes for this on the scale of tens of thousands of humans, which can be very difficult collaboration-wise and also in terms of cost. With linkage analyses in pedigrees, this is something that was done much earlier in the 70s and 80s when people were starting to do genetic studies, and are also kind of having a comeback as genomic methodologies become increasingly common. And these do detect bona fide and large effect variants. One of the most famous ones is Prader-Willi syndrome. This is a syndrome that causes obesity by preventing humans from having a sense of satiety. So they continuously eat to the point of harming themselves if possible. And these large variants are easily found using linkage analysis in families that have affected individuals. Of course, the resolution of whether or not we can detect a variant depends on the complexity of the pedigree. Very often in human populations, we aren't necessarily honest about paternity, and this can cause problems when conducting linkage analyses that are very awkward for the researchers. And these also can be used to detect relatively rare variants, but these rare variants may not necessarily be common to the entire population, so this can also cause some problems. Just to give you an idea of what's been done in the past insofar as obesity is concerned, these are all kind of the major GWAS studies, just looking at BMI and how that relates to obesity. For those of you that don't know, BMI, body mass index, is an index of height versus weight, and basically gives you relative mass for your height. And typically when we talk about an obese individual, we're talking about somebody who's in the top 5th percentile, so above the 95th percentile, of BMI within their population specifically. And that kind of definition is going to come back in a moment. So within all of these studies, you can see the sample sizes range from around 5,000 individuals to 125,000 individuals. And these are unique, unrelated individuals preferably. And then to replicate the results, the sample size get even larger. And you'll see some of the main genes popping up here include FTO, which we'll talk about in just a second. This is kind of the most famous obesity gene that we know of. And it causes the most variation in BMI that we know of. And then MC4R and MC4R is the other major allele that we know of here. And these are all implicated obesogenic genes or loci that we found using GWAS studies across the genome. Okay, so we've got a lot of loci that are implicated in humans at least. And these are based on a number of different phenotypes that we associate with obesity. Okay, so when we say obesity, like, we definitely have a mental image of what this means. But when it comes to actually measuring obesity, like I said, this is often a very kind of subjective and not very standardized measure. We've got this top 95th percentile of BMI, but people have also measured it in terms of body fat percentage, which is this orange circle, waist circumference, which is this crimson one, waist to hip ratio adjusted for height, which is the pink circle, and then just obesity as a phenotype, which is measured here in green. As you can see, we've got a lot of loci that are implicated only with one of these phenotypes, and then these major loci like FTO and MC4R that kind of are implicated across phenotypes within various studies. So again, this is interesting. We've got a lot of loci, but we don't really have a great idea of how all of these fit together, what the system's biology is. And then on top of this, these loci don't actually explain a whole lot of this phenotype that we're interested in. Okay, so if we're talking about human obesity, we've got about 40 to 70% of BMI at least being genetically controlled according to these studies, which is what we've got in this green square here. But the loci that we found only explain 1.45% of that variation in BMI. Okay, so that's like explaining maybe 2 pounds of difference between somebody who has these obesogenic alleles and somebody who doesn't. So we're still missing a lot of this variation and where that might be controlled from. And so if we're failing to find this using GWAS, then what do we have to do in order to make this more feasible for finding these loci? Where's the missing heritability? A lot of people argue that we need to look for more rare variants. So these are variants that have a minor allele frequency of less than 5%. So these are rare. In a human population, of course, with such large numbers, we would expect to find these loci relatively commonly. But again, still, it's less than 5%. So we need to look at the bottom 5% and the top 5% of these phenotype measures and kind of ignore the middle to try to focus more on individuals that might have these minor alleles. Another thing that we could do, and this is something that we're particularly concerned with at the Center for Neurobehavioral Genetics, which is where I'm working right now, is better redefining phenotypes. As all of you may know, the DMV, or is it DMV? Sorry, the Manual for Psychological Disorders.
DSM.
Thank you, DSM, DMV. Sorry, my car just got recalled. So. Don't buy a Chevy Cobalt. So the DSM has just been modified, and with that modification comes a modification in all these phenotypes that people are looking for with respect to bipolar disorder, schizophrenia, and these are phenotypes that people within my department are looking for genotypes for. So we know that we need to get better, more refined explanations or clinical signs or phenotypes in order to find genotypes that actually mean something in relation to the system's biology of these traits. So redefining phenotypes is kind of the project that I'm doing that I'll be talking about today. Finally, also relevant to this talk, there's an increased emphasis once again on pedigree-based studies, large-scale pedigree studies, that naturally exploit this increased probability of seeing copies of variants among those families. Okay, so this increases our statistical power with a smaller sample size, which is great if you've got the pedigree to work with. So, I kind of alluded to this before. Humans make a horrible model for studying obesity. In the past, we've relied on mouse models to kind of look for obesogenic genes. I'm sure everybody's heard of the ob gene, that obesity gene that causes this radical phenotype change within mice. And it's great, that work has been fantastic. Mice are a great model for studying obesity. However, the developmental period in mice is very different compared to the developmental period in primates. The life history in primates is much longer. We've got the longest juvenile phase and adolescence of any mammal. And so the growth patterns that we see and the fat deposition patterns as well aren't necessarily comparable, so they don't make a great model for understanding primate and human obesity. When looking at humans, we can't really control what people are eating. We can't really control the environments that they're living in. We can't rely on them to report honestly about what they're eating to us. So that makes studies of human obesity very kind of touch and go, because we can't control the environment in which they're placing themselves. And ultimately, longitudinal studies of humans are problematic because funding runs out after 5 or 10 years. On top of that, there's a very high dropout rate in longitudinal human studies because people move, they're mobile, or parents or children no longer want to give consent after a certain number of years because of some kind of change in attitude or mood about the study. So non-human primates strike kind of a great balance between these two different problems. Fat deposition patterns have been shown to be similar to what we find in humans. And on top of that, they've got this extended development that we also find in humans, not to the extent that humans have, but it allows for that kind of resolution during development that will help us understand why humans develop into obesity as well. So that's kind of all leading into what I'll be telling you about today. This is a study that's been done in conjunction with many people here at UCLA on the VRC, looking at the heritability of obesogenic growth in this Vervet colony that Lynn actually founded here at UCLA that has since moved over to Wake Forest University. Within that Vervet Research Colony, obviously there are vervet monkeys. This is a monkey that's common in across sub-Saharan Africa that were brought over to the Caribbean islands of St. Kitts and Nevis during the slave trade about 400 years ago, and then subsequently brought to the United States by Lynn and her colleagues in the 70s, and bred in the colony there. So we've got the pedigree going all the way back to, is it 1976, I believe? Some of it, yeah. And we've got them fairly well pedigreed and backed up with microsatellite data on how they're related with each other. Within this taxon, sexual maturity is reached at around 4.5 to 5 years, and they've got similar growth trajectories to humans in that they have this kind of growth spurt during puberty, and also sexual dimorphism, where the males are slightly larger than the females. Currently, there are around 350 pedigreed animals in the VRC, but the pedigree goes back much further, to around 950 animals. And 723 of those current and past members, we have whole genome sequences of. And these are at depth of between 4x to 50, or sorry, 40 to 40x. These animals are controlled in both their diet and environment. They're all fed a standard monkey chow diet that's controlled by the VRC. And they live in similar enclosures, about 16 different enclosures within the same environment. They have annual birth cohorts of about 25 males and 25 females per year, so that's wonderful for getting more individuals into your sample. And body composition and condition measures are taken about 3 times a year when they're tested for tuberculosis. It's great that they have to be tested 3 times a year because then we get these reliable measurements. And these measurements have been taken in the exact same way since about the year 2000. So now we've got a really nice longitudinal sample of comparable measures. So in starting this study, we just kind of took a look at the data that we already had to try to understand whether or not obesity was an interesting thing to look at in these animals. So I conducted a preliminary phenotypic analysis on about 657 animals for which we had data, about 410 of which were adults, so we could assess them on obesity measures. And these were only using the comparable measures from 2008 to 2011 when these animals first started at Wake Forest University, because I didn't want the move to be implicated in any kind of change in phenotype. And as you can see, these are the results looking at low S regressions across waist circumference. And this dotted line here at 40.5 is the line for obesity. This is the 95th percentile of obesity for waist circumference. And as you can see, with males in blue and females in pink, in the non-obese individuals, sexual dimorphism isn't really that radical. Okay? And on top of that, once they hit puberty around 5, growth more or less levels off. All right? But in those animals that were above the 95th percentile, you can see sexual dimorphism is exacerbated, and on top of that, growth continues, albeit at a slower rate than during development, long past they reach adulthood. Okay, so there's something different going on in the growth and development of these animals that become obese once they're adults, compared to the non-obese individuals.
Can I interrupt for just a minute?
Of course.
How are you defining growth? I mean, are you just defining it in terms of waist circumference, or are you defining it in terms of overall?
For this trait, we're defining it in terms of waist circumference, but when we look at the body weight information or mass, it's a similar pattern. This one just shows it more explicitly along the lines of how we're defining the obesity.
Okay, yeah, I mean, it's a different use of the word growth, so typically I would think we'd be talking about skeletal maturation.
Okay, yeah, this is only talking about body conditioning.
So they're getting fatter.
Yeah, they're getting fatter, exactly. Okay. So once we had these different trajectories, we decided to take these adult phenotypes and look for some kind of genetic signal or genetic heritability. So when running these across SOLAR, which is a program that we can use in order to get additive genetic heritability from a pedigree, using the VRC pedigree, we found surprisingly high estimated genetic heritability of these obesity-related traits. You can see waist circumference is about 61% controlled by additive genetic heritability, BMI around 65%, and those individuals that were chronically obese, that chronic obesity appeared to be heritable, 64% by additive genetic heritability. And by chronically obese, I mean these are individuals that were measured as obese 3 times in a row within the measurements, meaning that they were obese for a year or more once they had reached adulthood. So really high estimated heritability of obesity-related traits. So this led to the current study. So we decided, okay, obesity is important within this colony. It appears to be genetically controlled. Let's take a much closer look at how this actually develops. So what my goals were was I wanted to find an agnostic determinant of obesogenic growth. Okay, so I wanted to not just say if they get to above the 95th percentile when they're adults, then they're obese. I wanted to look at the actual developmental trajectories and divide them out into animals that grow to be large and animals that grow to not be large. And I wanted to see if this was something that came out statistically in the data. Once I had those, I wanted to generate growth models from which individual parameters could be extracted, so we can actually look and see what individual variation within these parameters might be controlled by genetically. So that also, of course, involves assessing the heritability of these obesity-related traits and their associated growth parameters. And then finally, using this whole genome sequence data that we have for the colony in order to find loci that would correlate with these obesity-related traits. Okay? So starting out with this agnostic clustering for growth trajectories, I used a k-means longitudinal clustering algorithm. This is one founded by Christophe Gentilini that you can use in R using the package KML. Using these longitudinal measures from the year 2000 to 2013 on individuals within the colony. What this program does is it basically randomly adds these growth trajectories within the model and then uses maximum likelihood to separate out these individuals into various clusters of growth, depending on how similar the trajectories are. The program also decides what number of clusters are most likely given the population data that you've got available, all right? Anywhere between 2 or 6 clusters based on how they fall out within the maximum likelihood algorithm. I analyzed the sexes separately because they are sexually dimorphic, so I didn't want that kind of interfering with the clustering algorithm. And what the program ended up giving was statistical support for only 2 growth clusters within each sex, 1 light and 1 heavier. And you can see over here, this is the clustering algorithm output for females. So 2 was the most likely number of clusters. In blue, we've got this heavier cluster, and in red, we've got this lighter cluster here. Okay. Within males, again, blue is the heavier cluster, red is the lighter cluster. As you can see, the males get cut off at a much earlier age than the females. This is kind of an artifact of colony maintenance. Within the wild, males disperse from their natal group, and the colony kind of recapitulates this by pulling males out of their natal group once they reach sexual maturity. Those males are subsequently put into other research projects or taken out of the colony, so the sample size for males is much smaller than that for females. So the results for females are much stronger within this study than for males. And happily, when trying to correlate these clusters with our kind of clinical definition of obesity that we used in the previous study, the chronically obese, in females at least, are significantly associated with the heavier growth cluster. Okay? Within males, that significance is not there, and I'm presuming that's because the sample size, once again, is much smaller within the males than what we have in females. So we've got this lighter and heavier growth cluster that's statistically supported without a predefined definition of obesity, which is great. The next step is to construct these growth models from which, oh yeah.
I'm sorry, can I just ask one thing that I didn't get about the clustering algorithm? So you said that it entertains between 2 and 6?
Yeah.
Does it entertain 1?
No, it doesn't. It automatically picks between 2 and 6 individuals. So yeah.
So how do you know that there's an actual bifurcation?
I mean, that's a good question. I mean, we're presuming that there is from the previous data that we've got some individuals becoming obese and some not. But yeah, statistically there's no way to kind of partition between 2 and 1, at least using this clustering algorithm. I mean, you go into it with an assumption that there is differential clustering within the group. And I think that we had pretty strong support for that looking at the data previously.
Okay.
So constructing these growth models, we used a logistic growth curve in a nonlinear mixed effects model using LME4 within R. The logistic model looks more or less like this. Theta 1 is the asymptote of growth. This is the maximal weight that you find within the animal. Theta 2 is the midpoint of growth, so this is a measure of basically how long they're growing for. And theta 3 is the rate of growth, okay, so how quickly they grow. Age and parameters of growth were set as fixed effects, and then sex and cluster, so male heavy, male light, female heavy, female light, and individual ID were set as random effects. What this kind of model allows us to do is pull out not only these fixed effects parameters, so we can get an idea of what the average parameters are for this whole population, but using these as random effects, we can control for individual and pull out individual parameters. And on top of that, we can also look at gross differences between sex and weight cluster that's defined in this agnostic clustering algorithm. So you can see how different those growth parameters might be between those groups as well. So these are the results of these models. In dark blue, you've got the heavy cluster of males. In light blue, you've got the lighter cluster of males. In dark yellow are the heavy females. And in light yellow, we've got the light females. Okay? So as you can see, they're separating fairly well. This is the asymptote of growth here, represented again by theta 1. And the colors are consistent throughout, so light females, heavy females, light males, heavy males. You can see obvious sexual dimorphism. The males are pretty much always heavier than the females once they reach adulthood. But we can also see that there's a definite difference between these two agnostic clusters of growth, where these light females are becoming lighter in adulthood, and the heavy females are becoming heavier. So this is also kind of supporting this kind of dichotomy in growth within each sex. You can see the same kind of difference within males, although it appears exaggerated. Again, the sample size is very small for males, so I wouldn't make anything of that. Theta 2 is, again, the duration of growth. We know that in most primates from previous studies, that when we have sexual dimorphism between males and females, this is often reached through what we call bimaturism, meaning that males, instead of growing more quickly than females to reach that larger adult size, they actually just grow for a longer period of time at the same rate. And these results seem to be recapitulating this. This midpoint of growth is higher within females. Think of it this way, like a lot of people are like, oh, why is it going that way? By having a higher midpoint of growth in females, that suggests that the overall duration of growth is actually shorter. Okay? So females are growing for a shorter period of time in the light category than the heavy ones. So this is suggesting that these obese animals, these animals that become heavier, are becoming heavier because they're putting on weight for a longer period of time consistently than these lighter females, which seems to make sense. We've got a similar pattern within the males as well. So obesogenic growth is more an issue of growing longer into obesity than growing heavier more quickly. Okay. And then finally, when we look at the rate of growth, in females where we've got a really good sample size, we see that the rate of growth is actually not different at all. Okay? So body weight, these obese animals aren't growing fat quickly, they're just growing longer into their obesity. So this kind of recapitulates again what we're seeing in that first graph, where this body condition growth is increasing over a longer period of time, not necessarily faster. And like you said, with skeletal growth, I wanted to look at crown to rump length, which is a better kind of proxy for skeletal growth instead. And we actually have somewhat of a similar difference between these animals. So there is actually a skeletal difference in growth between these obese versus non-obese animals as well. So these obese animals are actually skeletally significantly larger than what we have in the non-obese, and this is the same across both sexes as well. But as you can see, the separation in growth patterns is actually not that different. Okay? So what this is suggesting is that these obese animals are also somewhat larger, but they're not developing into this largeness. This seems to be much more genetically controlled. So to look at this genetic control, again, we want to find the heritability of all of these growth parameters within these species. So we use the VRC pedigree. This is just a small snapshot of it. As you can see, it's kind of a mess. So thankfully, we can just plug it into Solar and let Solar take care of that mess for us. For adult traits, similar to what we did in the preliminary analysis, I averaged traits across each individual. So this is just looking at static adult levels of waist circumference, BMI, body weight, etc. Because it does fluctuate over time, but around a specific mean for each individual. With these growth parameters, we estimated them for each individual out of that mixed effects model and extracted theta 1, theta 2, and theta 3 for each so that we could run them across Solar. And the phenotypes were analyzed using maximum likelihood variance components analysis using that VRC pedigree within Solar. Okay, and this is what we end up getting. So using these new agnostic clusters and the increased database, we end up getting actually much higher heritability values for all of these static obesogenic obese traits within adults. So body weight is about 89% genetically heritable, waist circumference 81, BMI 77. Crown to rump length is 60, which is less than what we have for the body condition values, which is interesting. And then this chronic obesity is about 89%. So these are extremely high values, much higher than what we get in other studies of obese non-human primates, including previous studies within the VRC. And this to me suggests that this method is finding much more of the variation that is implicated within obesogenic traits. When looking at the growth parameters, slightly lower heritability, but still in the high range. We've got 71% for this asymptote of growth. This is about what we'd expect, because this is like looking at adult body size, so we'd expect heritability to be that high. Looking at duration of growth, we get around 69%, which is also very high, and that's great. So it's suggesting that these animals that are growing into becoming much larger, heavier animals, this is largely genetically controlled. Okay, this isn't a matter of them eating more, unless eating more is also genetically controlled, which is something that we have to think about. And then finally, the growth rate is actually not very significantly genetically controlled at all. And we would also expect this looking at other primate studies that find that the rate of growth isn't so much genetically controlled as mediated by environmental factors. Interestingly, in crown to rump length, the heritability values are a bit smaller, but again, they are both very high for duration of growth and asymptote of growth, and growth rate itself doesn't seem to be genetically implicated insofar as the other traits. So finally, the last step of this process is to look for loci associated with these parameters and these traits. And we've done some preliminary association analysis of mean adult BMI using the pedigree. We inverse normal transform them, regress them on age and sex within SOLAR, and then used MX to look for actual SNP hits within all of the contigs that we have. Unfortunately, the reference genome is still being annotated right now, so I can't tell you if this is FTO or if this is MC4R or what. But we did find about 34 independent SNPs that seem to be implicated in variation in BMI across this population. With p-values between 10 to the minus 4 and 10 to the minus 5. Now, typically with genome-wide association like this, you want a 10 to the minus 8 to know that it's genome-wide significant. But we're pretty confident that we've done a power analysis, and we know that if we just increase the sample size with these increasing birth cohorts in animals that are aging up into it, that we should be able to get genome-wide significance shortly. And just to give you an idea of how wonderful this data looks to us, at least, this most significant hit on contig 37, we looked at the number of C alleles within that contig and found a pretty nice separation with 0, 1, and 2 C alleles, suggesting that this C allele variation within this locus is implicated in adult levels of BMI. So we're pretty sure that this hit is having a very real impact on BMI within the adults. And we're hoping that, again, with increased sample size, and when we finally run the multi-point linkage analysis, which has better power to detect these, that we will have some very nice obesity loci to work with after the whole analysis is done. Okay, so what we get out of this then is that adult onset obesity is in part a developmental process that's under genetic control, because this is an extended kind of form of bimaturism within each sex that's leading to these obesogenic profiles. There's a sex component to obesogenic growth, and we need to do more analysis to kind of parse out how different the values are between males and females and whether different loci might be implicated. And then finally, multiple loci are implicated and also detectable within this model. So using this model, we should be able to get a number of loci that are implicated in a number of different phenotypes that are associated with obesity. And what we're hoping is that we can translate this into information for humans that could help kind of detect risk factors for obesity in humans, which is a long way off, but that's what we say for the grants. So what's going on next with this information? The short answer, within the VRC, obviously we want to look at age and sex-specific heritability and also find QTL that are implicated with age and sex-specific data sets. Obviously further SNP discovery for growth phenotypes, and then multi-point linkage in SOLAR with WGS samples so that we can actually get these strongly supported loci. And then the long answer is obviously we want to look at proximate aspects of obesity, like what's the systems biology that leads to these different phenotypes within the individuals. And we also want to look at ultimate aspects of obesity, which would require further field work. Now we know that in the field, studies of complex traits in wild non-human primates have been really, really difficult to conduct. Okay? These are often precluded in these populations because the studies haven't been going on for long enough. All right? A lot of studies have only started in the last 10 to 15 years, and so we don't have really a depth in the pedigree to be able to detect these kinds of differences. So GWAS isn't possible because sample sizes are too small. Linkage, these wild pedigrees are often temporally shallow or incomplete or uncertain. But we do know that it's becoming increasingly possible. For example, at Susan Albert's field site, Jenny Tung is doing a lot of work with baboons, trying to incorporate whole genome analyses within that population with some success. And in other non-human animal species, it's actually becoming increasingly feasible. Some really exciting work is being done by Maya Tarka, who's in Bengt Hansen's lab at Lund University, actually looking at fitness values for traits that they've been able to do genome-wide association analyses on. So it's very exciting. It's a great model for what is possible once we use the colony to get these loci, and then we can look for these loci within the wild. So the way that we're trying to do this, and this is kind of a shill for the research group that I'm in, I'm part of something that's been variably called the International Vervet Research Consortium, or the Vervet Genome Project. It's had a number of names. But it all is kind of bringing together this larger species-wide model, or taxon-wide model, so that we can actually get an idea of what is happening within the wild within these populations. So we've sampled from multiple populations within mainland Africa. We've sampled extensively within this bottlenecked population in St. Kitts and Nevis. These are all sample locations that we've done on the island. And then obviously we've also sampled from the VRC, which was what this work has been talking about. Within these collections, done jointly with UCLA and the University of Wisconsin and several other institutions across the world, we've managed to collect information on obviously over 950 animals in the VRC, about 700 animals within St. Kitts and Nevis, collected from the wild and from a captive colony there on the island. And also within Africa, we've managed to collect from over 1,000 individuals within the wild. Within captivity, we've got a lot of different phenotypes that we are trying to work with that could be implicated within this study further. So we've got phenotypes on brain and behavior. We've got metabolism and growth phenotypes, which will be more significant for this study, obviously. We've got information on microbiome from multiple body sites, which also is becoming increasingly important to studies of obesity. I don't know if you guys are familiar with the microbiome work, but we found that the ratios of certain bacterial families within the gut actually shift significantly with BMI differences in humans and other non-human animals. So this may be implicated in some way that we might be able to parse out within this colony. And then of course we also have EQTL, or expression QTL, where we're looking for loci that control for various traits based on gene expression profiles. Okay, so we've got a really nice, rich phenotype database within this captive colony. Within the wild, we've got a lot of similar values. This is an example of the type of trap we were using. These are kind of modified drop traps that we hauled across mainland Africa. And these are animals that we've captured in the wild. We put them all back. We don't sacrifice them. So these are all phenotypes that we can collect within the half hour that we have them under ketamine sedation. Within this, though, we've managed to also collect a really rich phenotypic database. We've got cerebrospinal fluid in some populations, DNA, RNA, tissue, plasma serum, dental casts, many, many other traits, including measurements. And all of these animals have been tagged with a microchip so that if we retrap them, we can actually identify them again. So this gives us the possibility of doing longitudinal studies within these wild populations as well, which is what I would hope to do next. So looking at kind of the systems biology of obesity, we could use these loci that we detect that vary for BMI to select individuals for kind of cohort-based research into obesity interventions during development. So this would be kind of like pseudo-knockout animals, okay? We also are really interested in doing research into maternal effects on obesity. Like I said, we already know that this happens in humans, but within the VRC, we actually have evidence during a dietary intervention that those animals that were gestating during a dietary intervention, during which time the animals were put on kind of a typical American diet, those animals haven't reached puberty yet, but their growth trajectory is almost vertical compared to the growth trajectories of the other animals. So we know that there is possibly some kind of maternal effect, or at least we're fairly confident that we're going to find one once these animals age further, and we can actually look at the differences in developmental trajectories. Obviously, dietary modification studies are possible within this population, so we could do gene by environment interaction. And then we've also done actigraphy studies kind of preliminarily within the colony. And we have found significant differences in various individuals in their actigraphy assessments based on putting kind of an accelerometer onto their body and watching them move around. And we know this has been validated in the wild for various primate species as well. So this could lead to very interesting studies comparing the captive and wild populations as well. Biomedical aspects that we could translate this to. We've got fibroblasts available for almost every animal sampled. So we could actually do cell cultures and look for actual cellular level differences that are implicated in these loci differences. So we can kind of understand, again, what the systems biology is of these genetic differences. We have data for RNA-seq, so we could look at gene expression pathways and again come up with a better idea of what's going on system-wide within these animals that's causing obesity at different developmental phases. And again, we could have potential extension to a mouse knockout model if QTL have mouse orthologs. Okay, so in this case, we could actually do this research much more quickly within a mouse model. I still think the primate model is much better. And then finally, what I'm most interested in, and which I think would be most interesting within anthropology at least, are these ultimate aspects of obesity. So what we're going to be doing next, and I actually have just been offered a postdoc at Berkeley in order to conduct some of this work, is a preliminary search for variation in these discovered loci within the captive colony to look for variation within the wild, okay? So we would be doing kind of a population genomics assessment on these wild populations in order to find out if there's any kind of environmental variability that leads to selection for these different obesogenic genes. This is important because we have this idea of how obesogenic traits have evolved in humans, okay? There's two different kind of schools of thought. We have this idea of a thrifty phenotype, okay, which is the idea that if you're developing within a certain type of environment, then maternally you're programmed to expect that environment, all right? So if you grow up in an environment where there's starvation, then you would be expected to develop a phenotype through some kind of developmental programming or gene expression variation that would lead you to crave the foods that are lacking so that you can eat as many of them as possible because you know they'll be scarce, okay? There's also the thrifty genotype idea, which suggests that these genes are static and that they're selected for by kind of feast-famine cycles within your evolutionary history as a species, okay? You can actually parse out the difference between these two hypotheses using gene-by-environment interaction studies and selection studies just based on the genomes of animals that have these traits, okay? And both of these are going to be possible given the larger framework that we've developed within this International Vervet Research Consortium. So instead of just hypothesizing about these ideas, we can actually go and test ideas in the wild about how these obesity-linked loci actually may be selected for in different environments. So this would be kind of the first test of that kind of genetic framework for obesity that's been conducted in the wild. And that's all I've got. So if you have any more questions, please ask. If you want, I can slip back through slides or talk more about the model or the analyses. I'll be happy to do so. Thank you.
Yeah. So I'm glad you mentioned microbiome there toward the end because one thing that I was thinking about in here is when you're looking at heritability in general, the possibility of vertical transmission of microbiome rears its head is one potential explanation. Now, since you have paternal relatedness to calculate in as well, my first response to that is, well, that's okay. We can take care of that because we can look at the paternal contributions. But it seems to me that there's another possible explanation for a pattern that might look very similar to that one, and that's if you have paternal imprinting going on, where you can get different phenotypic effects based on whether the given allele is being inherited maternally or paternally.
Yeah.
So how do you differentiate between the gene-by-environment interaction where you have vertical transmission to microbiome and a paternal imprinting explanation? Because phenotypically it seems to me that they produce the same pattern across the pedigree.
Yeah. It's a really good question, and it's one that I'm totally unequipped to answer right now. But that's something that I would want to bring in a collaborator on who has more expertise with that information so that they could actually analyze that further. I mean, like I said, I'm trained in anthropology, and like quantitative genetics is about as far as I go. And this is part of the reason why I'm going to be going to Berkeley. I'll be working with people in Monty Slatkin's lab, and actually somebody in particular who's interested in studying the microbiome, like kind of parsing out this microbiome heritability versus the actual like autosomal genetic heritability that we would expect to find in these animals. So yeah, I mean, like it's definitely something that we need to look into. But I can't.
You can experimentally manipulate it in captivity.
Within captivity, yeah, like yeah, by doing like microbiome replacement studies and separate housing studies. Yeah, exactly. Which is something that I would like to look for a collaborator to work on.
I mean, I have no idea whether people have thought through. You might need modeling to do this. I don't know, but what the implications of sex bias dispersal are for imprinting. But you can imagine a sort of back of the envelope sketch in which if you're a female and you're going to be growing up around and then living with your same-sex kin, right? That the trade-off between selfish behavior and nepotistic behavior is different in terms of your monopolization of food resources than if you're a male, where you're going to grow, get big, and then leave, right? And then subsequently you're not going to be competing with those same individuals. The trade-offs might not be the same, so you can imagine a situation where that would favor a process of imprinting. But it's way beyond my camp.
Yeah, me too. And obviously, working within this single species model, we wouldn't. I don't know how well we'd be able to address it. I mean, it would be better to look at variation across animals that have different dispersal regimes, but are still somewhat closely related, which would be somewhat possible in primates. But we don't have the data or financial resources to do that within this model, at least. Like something like a rodent model, which would be more appropriate. And that's actually the collaborator that I'm hopefully going to be working with on this, is looking at rodent variation across South America in microbiome. So he's bringing in that expertise, and hopefully he'll be able to address concerns like that. But you're right. I mean, what is it? Studies have shown recently that the gut microbiome is actually transmitted via breast milk. So yeah, definitely vertical transmission of the maternal gut microbiome. But paternal, it'd be interesting to look and see how similar that might be.
And I mean, there's some evidence that animals in the wild follow cohort patterns of obesity that track obesity in human populations nearby, and that's true even when you control for sanitation and refuse disposal and things like that, which suggests that the humans are, populating the world with some obesogenic, endosymbionts, and they're spreading them to other species.
To all animals that are associated. Yeah. So it will be very, very interesting to look into.
Yeah, I mean, just following up on that, it seems like, in addition to what Dan was saying, all kinds of epigenetic effects are going to be hard to tease out, because if you're estimating the genetic variance from the pedigree, that's confounded with anything vertically transmissible, right?
Yeah.
So it could be that those heritability estimates are
inflated.
overestimates.
Yeah, no, I mean, that's a concern that I have too, because they're so much higher than what I would expect, and that I think what other people would expect. And so, yeah, I mean, I was asked this recently, and I don't have a good answer for it, because I don't know how I would parse out a microbiome effect
I don't think anyone does. That's why this is a problem, right? And
yeah, it is. I mean, it makes me wonder about previous studies that have looked at heritability of obesogenic traits as well, because obviously, like, we know increasingly that microbiome is having a huge effect. Like, when they've got this off-balance ratio of bacteroides and firmicutes, which are the two big ones that are kind of implicated in obesity, we know that in humans at least, they actually metabolize their food much more efficiently when they're obese, which kind of leads to this positive feedback loop of becoming more obese because they're getting more energy out of their food. But I mean, like, how do we actually quantify that in something like a additive genetic variation type framework? I have no idea.
In the early work at the colony, we included a maternal effect in
Mhm.
solar models where you're just including a component that has maternal ID.
Yeah.
And I don't know enough about how solar works, but I think the people that build it are biased against environmental effects.
Yeah.
And it's amazing how many traits you put in where there's no maternal effect. But Julia Bailey did find a maternal effect, a significant maternal effect for body weight.
Okay.
Which is in the model, above and beyond the genetic contribution. And I'm not sure what's happening inside the black box. If they're comparing the, through the paternal line and the maternal line.
Yeah.
But for body weight, gestation and lactation and these other kinds of vertical transmission really probably do have an effect. And if you just run the solar, it does inflate the heritability if you don't account for it. But still, even accounting for the maternal effect, the H squared, we're still about 0.6. So
Yeah, it's still very high.
So one other thing I wanted to ask, in the human picture of that unfortunately obese child, it does seem with the monkeys that none of the juveniles were even what one would call fat.
No.
It wasn't until they got to adulthood. Do you think that's a difference between, or it's just that human populations have gotten to such an extreme state that monkeys haven't arrived at that point yet?
Yeah, it's a really good question. I mean, you can see here too, like, by definition, none of the young ones were obese, and there's no evidence of any kind of real difference during development there. And we do find in human populations that there are people that only become obese once they reach adulthood. And it does seem that, functionally at least, this kind of juvenile obesity is a different kind of phenomenon.
Yeah.
And so that's something that we need to work on too, that this model I don't think can really address. Like, I don't think we can talk about childhood obesity within the Vervet model. Although, again, looking at that typical American diet cohort would be interesting because, like I mentioned, like if you look at the profile, they would be growing like this. So they are becoming heavier at a much earlier age, but it's only one cohort of infants, and so I'm not sure what we'll really be able to get out of that. But yeah, you're right, like, this can't address childhood obesity. This only addresses obesity upon reaching adulthood. But yeah, we need to come up with some kind of functional definition between these different types of obesity as well, if we want to actually get at what the biology of what's happening.
Because it's so unusual in any species to see a fat.
A fat baby, yeah.
Well, just an observation from my parents' yearbooks. I can see that they grew up in the Depression, and there was no obesity back then. Out of yearbooks with hundreds and hundreds of kids, they're just all seen.
Yeah.
It was.
I mean, it raises a lot of interesting questions about how this could develop. I mean, if we talk about thrifty phenotype, it might be a kind of positive feedback loop on an evolutionary scale where you've got a larger mother that's priming a larger offspring that's priming larger offspring in turn.
So something's happening, but it's it'd be certainly a source where you can get a large body of data, and you just raise metrics where you could apply to those types of images. And because yearbooks, you could get hundreds of thousands of populations of hundreds of thousands of post-puberty kids and deal with it statistically. But I can tell you, back then it wasn't.
Yeah, I mean, that would be interesting if you could use like photogrammetry. I think it was what you're talking about to look into that kind of thing. I know with longitudinal human populations, like the Fell's longitudinal study is also tackling some questions like this, where they've got thousands of individuals over developmental periods. But I haven't seen the results from that come out yet. But yeah, very, very interesting.
I don't suppose there's any chance with the vervets that you could do in vitro fertilization and have females gestate infants that are not their own congener theirs.
I don't know, Lynn, is that something that we could do?
Nobody around has ever really had the expertise. It would be more possible to cross-foster for later effects because they're very interested in caretaking others' infants, and you can get adoptions. But, I don't know, the people in Oregon are doing that. So it would be possible if there were colleagues who were understanding this.
So, Dustin Penn and Wayne Potts work on rodents, and they have, I would say one of their major contributions that the Potts lab has made is to show the gross underestimation of the size of the effect in breeding depression when you have laboratory animals versus animals in something remotely resembling their wild state. So Wayne Potts has this giant barn that is mouse land where the animals are free-ranging and they compete. There are no predators in there, but there is competition.
Yeah.
And you get just dramatically increased effects of inbreeding depression there. And I'm raising that as an analogy by way of saying, well, so yeah, we never see obese vervets in the wild, right? To the extent that we want to discount all the other possible explanations and say, fine, there are obesogenic genes, which seem plausible. Then you're back to the ultimate question of what are they doing, right? And I wonder how much we're being deceived by the zoo-kept nature of the population in terms of misidentifying what the phenotypic consequences are, because since we never see them get fat in the wild, if those genes are there, they're doing something, right? What are they doing? Well, it's not clear that you'll be able to get to that in any kind of clear sense from a population that's sheltered from the selective pressures that might reveal the adaptive function of those variants.
Right, and that's why I'd really like to take this study back to the wild. I mean, I acknowledge that like a lot of people ask this, which is interesting, but I don't expect to see any obese monkeys in the wild. Like I don't expect that at all. But I do expect to see these obesogenic alleles being variable across environments. Like I would expect them to provide some kind of advantage depending on the environmental context, because they're doing something that's leading to an increase in bulk. And how that's actually accomplished, I mean, I can't tell. I'm just looking at the phenotype right now. Further studies within the captive population, looking at how this is implicated in, I don't know, satiety or impetus to eat or appetite. I mean, this I can't say yet. But what I do expect is for whatever this is controlling for to offer some kind of variation in body condition in the wild that I think could be selected for. Interestingly, like a lot of people have also asked about the idea that this is a highly inbred population, what the linkage disequilibrium is like what we would expect to find in the wild because of that. But because of the kind of layout of this study, we do have ways of addressing that. Like we do know what the difference in linkage disequilibrium is between the VRC and St. Kitts and then the rest of the population within Africa. So I feel like we're a lot closer to being able to address that, that kind of question. But until we get more information on what's happening within these wild populations, like it's necessarily speculative. And it's for somebody else to speculate, because I don't think my expertise is quite so good to discuss that.
Yeah, I mean, one challenge, it's great that you're taking it to the wild. One challenge is that the selective advantages may be manifest only in infrequent events, right?
Yeah.
So you can easily imagine a situation where this counts when there's a famine, for example,
exactly.
or something like that. And that only happens once every decade or something, where there's a strong selective filter being applied, but then it really counts.
Yeah, well, I think we're lucky too in that the sampling that we've done across these different regions, sorry, I'm like jumping through all of my slides. I think that's one of the advantages of the sampling regime that we've done, because within the South African population at least, we've got a variety of different environments. So within the Hari Dam population, for example, they live on agricultural fields, like corn and wheat. So I would expect there to be like much higher body quality indices there, heavier weights, not obese necessarily, but heavier. Whereas in Seduring and Sandveld up here, these are game reserves where in Seduring there is some provisioning because there's people that go picnicking there, but for the most part they're kind of foraging on their own in kind of these scrubby riverine environments. And then in Sandveld, which is a little bit further north, there's absolutely no provisioning happening at all. So I think that we could expect to see at least moderate differences within these populations, where we could kind of parse out these kinds of effects. As for how long, how far back in time that environmental impetus has to be in place before we could actually detect something, I'm not sure. But I do think that we have kind of environmental variability built into this model that we can use to address that kind of question.
Yeah, even seasonal variability, I think, could promote strongly seasonal variability in food quality, could promote the maintenance of these genes, which we call obesogenic, but really are helping you, keep weight.
But
In the Karoo Desert, where I was last summer, between those 2 points, it's really an extreme habitat. I mean, I'm used to seeing vervets, eating fresh pea pods from the acacia trees. Well, they were rooting through the ground and finding like an old dried up pea from 3 months ago and popping it in their mouth at the end of the dry season where there's no standing water. I mean, it's really pretty amazing what they put up with. They looked fine. I mean, they looked great. Nobody even looked skinny. So they probably have these genes that we're decrying in humans.
Yeah.
I think that would be a good place to look.
Yeah,
Include Peter Hainsey's site in your study.
Okay, yeah, I'm definitely going to get in touch with them.
So I mean, in order to maintain variation in the population, you have to have one of two things. So either the animals have to be moving, there has to be plenty of gene flow between, extremely Spartan environments and relatively abundant ones, or the time scale of the variation has to be longer than seasonal. Because if every year there's a hungry season and then there's a season of abundance, and the obesogenic genes are an asset, then it should go to fixation. Everybody should have them, right? But if they're, famines every decade, then the costs of those genes that get you through that bottleneck may be high the rest of the time. You may be at a competitive disadvantage for other reasons, right? And so that's going to maintain variation in the population. So, I think it's useful to sample from these Spartan environments precisely to see whether you see differences in the frequency. But unless the animals are frequently moving between ecosystems, that alone can't explain the maintenance of the variation.
Vervets are fairly present throughout every place I've been in South Africa. I don't know if there's big barriers or the populations are relatively continuous.
Well, we can address that with the whole genome sequencing also. We have about 120 animals, whole genome sequenced from these populations. And we do see evidence of gene flow going from the north to this area and the north along the coast, and with very little gene flow going between these two populations. So we actually could maybe predict the direction that we would expect this gene flow to occur. As we're doing it on like a population basis, I don't know, I think that we maybe could address that with this kind of wide-scale sampling as well. Yeah.
Just following on, I work up in northern Namibia, and there's vervets up where I work, but I'm just thinking they would be an interesting dot to add to your map because there's, of course, major desert between, so there probably is not any gene flow between the colonies where I work and the rest of these, so they would be sort of an interesting comparison to, against these guys who may have more gene flow.
Yeah, definitely. Looking at kind of the larger map, we've sampled extensively. We've mainly sampled in this region, up here in West Africa, and then in East Africa, and then we've got other populations here, here, and here that we've sampled from. But yeah, it would definitely be interesting to see a broader array and look at how gene flows work within those populations, because we still don't have a good idea of what the phylogeny of these. We don't even know if they're species. Trudy Turner, who we work with, is they're not species, they're morphotypes, because we don't actually know that these are different species, and this is something that we're dealing with as well. So yeah, sampling from more populations is something that we definitely should be doing.
And just the fact that this typing came from the animals, the small number, some unknown small number of animals that got the same kits in the 1600s, but there's still two types around, might suggest that there's little harm in this variation, little, less effect
in
of
the
the
wild,
variation, so it's not a disadvantage until you get into such a ridiculously food-rich environment that it can actually create problems.
Yeah.
But there has to be some cost to it if there's a benefit, say surviving famine or dry season scarcity or whatever. Because otherwise, if there's a benefit and there's no cost, it'll go to fixation, right? That is, you'll lose variation.
But if you
population.
never, if it's so rare to achieve the benefit,
It just, I mean, depends how long the system runs and what the extent of the benefit is.
Well, this is another reason to like go and actually look at wild populations and how they're developing over time, because I would like to find out what kind of developmental plasticity is associated with these different loci. I mean, if they are actually developing differently depending on their genotype, then there may be some other selective force that's implicated other than nutritive or survival benefit, like maybe a different body condition, I don't know, looks different in the wild than what we would expect it to. I mean, this is totally speculative, but this is another reason why I think we need to go back to the wild and see how these genes are playing out phenotypically. Because I think like it's decent to assume that this is going to be a body condition impact, but we don't know like what the system's biology is, so that like until we actually see what the difference looks like in the wild, if there is one, like it's all speculative.
yeah, it could have something to do with activity level.
Yeah, exactly.
Something like that, where there could then be a balanced polymorphism within a population. It's easier to understand. Anyway, good idea. Also, this notion of getting away from this is the bad gene variant and this is the good gene variant, you know, I mean, as an anthropologist should.
Yeah, exactly. I don't want to, that's something, this is the fat gene. Like, it's
Yeah,
not
right, exactly.
necessarily the fat gene, it's just the
The
gene that
biomedical
does something.
approach.
Yeah, exactly. Yeah.
I'm just wondering if at the VRC, if they mimic, so if there are these kind of seasonal patterns of scarcity on an annual basis, if that's something that gets mimicked in the diet at the VRC, or they get sort of the same food every day?
To my knowledge, they get pretty much the same diet every day unless there's an intervention.
Okay.
Yeah.
Because it'd be interesting to see if you put them on a diet that more closely mimicked those seasonal patterns, if that has long, you know, I mean, you probably would have to do it for multiple years, but.
That was somewhat seasonal, but not enough. Still, the chow was the basic food.
Any other questions? Well, thanks for your attention, and thanks for the great discussion afterwards as well. This has been fantastic. And I think, what is happening next? Like food, coffee?
Food's next.
Yeah, so if you want to keep talking, we can keep talking. Is that